Deep within the folds of the human brain, a sophisticated biological navigation system creates a living map of the world. This hidden architecture guides every step you take. The primary structure responsible for this feat is the hippocampus, which functions as a dedicated processing center for spatial information and memory. Research in neuroscience has shown that this region does not just store facts, but actually constructs a three-dimensional representation of your physical surroundings. This mental model allows you to navigate through a dark house or find your way home from a distant city without needing a physical compass. It is a biological marvel that operates silently in the background of your every waking moment. If you enjoy these relaxing facts, please consider subscribing to the channel. The brain achieves this by constantly calculating your position relative to fixed landmarks in the environment. This process, known as self-localization, relies on a continuous stream of sensory data from your eyes, your inner ear, and even the tension in your muscles. Scientists have discovered that the brain maintains this internal coordinate system even when you are sitting perfectly still or resting. This persistence ensures that when you open your eyes, your sense of place is immediately restored and synchronized with the physical world. The complexity of this system is evident in how it handles the vast scale of the environments we inhabit daily. Unlike a paper map, the brain's version is dynamic, expanding and contracting its resolution based on the importance of the location you are currently exploring. A familiar kitchen might be mapped with incredible precision, while a long stretch of highway is represented by a more generalized, efficient set of neural markers. This flexibility prevents the brain from being overwhelmed by unnecessary data while ensuring you never lose your sense of direction in critical moments. One of the most remarkable aspects of this inner GPS is its ability to function through pure intuition. Even without conscious thought, your mind is performing complex geometric calculations to keep you oriented within a room. This effortless awareness is what allows for a peaceful sense of presence in any environment you visit. Sharing these science for sleep insights helps us appreciate the intricate machinery that keeps us grounded. By understanding how the brain tiles the floor of our reality, we begin to see the profound connection between our physical location and our very sense of self. Deep within the folds of the human brain lies a structure so specialized and elegant that it serves as the literal foundation for our sense of place. This region is known as the hippocampus, a name derived from the ancient Greek words for horse and sea monster. When early anatomists first glimpsed its curved, tapering form, they were struck by its remarkable resemblance to a seahorse. This delicate structure is tucked away in the medial temporal lobe, positioned symmetrically on both sides of the brain. Its physical placement is no accident of biology, as it sits at a crossroads where sensory information from the entire body converges to be transformed into a cohesive map of our surroundings. The discovery of this region dates back several centuries. Originally identified by the Venetian anatomist Giulio Cesare Aranzi. In the 16th century, Aranzi observed the unique ridges and curves of this internal landscape and gave it the name we still use today. At the time, the true function of the hippocampus remained a profound mystery, hidden away from the rudimentary tools of early medicine. It was viewed primarily as an anatomical curiosity rather than the sophisticated engine of navigation and memory we now know it to be. Over the following centuries, scientists slowly began to peel back the layers of its purpose, moving from simple observation to a deeper understanding of its cellular logic. The architecture of the hippocampus is unlike almost any other part of the central nervous system. It is composed of layers of densely packed neurons arranged in a distinct, interlocking pattern that looks remarkably like two C-shaped structures gripping one another. This specific layout is known as the hippocampal formation, and it includes several subregions that act as a high-speed processing circuit. Information enters through a gateway called the dentate gyrus, where it is sharpened and organized before being passed through a series of fields known as CA1, CA2, and CA3. This rhythmic flow of electrical signals allows the brain to take raw, chaotic data from the outside world and turn it into a stable, enduring representation of a physical room or a winding forest path. For a long time, the scientific community believed that memory was scattered randomly throughout the brain like grains of sand on a beach. It was thought that no single area held the master key to our past or our spatial awareness. However, modern neuroscience has fundamentally overturned this belief by demonstrating that the hippocampus is the central hub for spatial mapping. While early theories focused almost exclusively on how we remember names or dates, we now understand that our very ability to navigate a house in the dark is tied to these specific cells. This shift in perspective has revealed that our memories are not just abstract lists of facts, but are instead deeply rooted in the physical geometry of the places we have visited. The transition from viewing the brain as a simple recording device to seeing it as a dynamic mapping system has changed how we define the human experience. We now recognize that the hippocampus does not just store the what of our lives, but more importantly, the where. This evolution in scientific thought highlights how our internal world is built upon a literal physical grid. By exploring these relaxing facts, we can appreciate the quiet, steady work being done inside our minds every single second. This sophisticated seahorse-shaped organ continues its work even now, maintaining a steady, silent vigil over our internal sense of location. This constant mapping provides a profound sense of continuity, ensuring that we always know where we stand in the vastness of our world. Deep within the quiet architecture of the temporal lobe, a specific type of neuron serves as the primary anchor for our sense of location. These remarkable units of the brain are known as place cells, and their discovery fundamentally changed how we understand the intersection of biology and geometry. In the early 1970s, a neuroscientist named John O'Keefe began a series of experiments that would eventually earn him a Nobel Prize. He was interested in how the brain processes the physical world, moving beyond simple sensory input like sight or touch. By recording the electrical activity of individual neurons in the hippocampus, O'Keefe stumbled upon a phenomenon that seemed almost like science fiction at the time. He observed that certain neurons remained completely silent for long periods, only to burst into rapid electrical activity when an organism moved into a very specific spot in its environment. This was not a response to a specific object or a flickering light, but rather a response to the location itself. Each of these cells acts like a tiny biological pushpin on a mental map. When you stand in your kitchen near the stove, one specific group of place cells might fire. When you move toward the refrigerator, those cells go quiet, and an entirely different set of neurons takes over the responsibility of signaling your position. The mechanism behind this is incredibly precise and consistent. Scientists often describe this process as tiling an environment. Imagine walking into a brand new, empty room that you have never seen before. As you take your first few steps, your hippocampus immediately begins to recruit available neurons to represent different patches of the floor. One cell might claim a small circle of space near the door, while another claims a square meter in the far left corner. Within minutes of exploring, these cells have effectively tiled the entire surface area of the room with overlapping fields of neural activity. This creates a seamless internal representation of the space that allows you to know exactly where you are relative to the walls. This discovery was first visualized through careful observations of rats exploring simple circular arenas. Researchers watched as the animals wandered curiously around the open space, sniffing the edges and moving toward the center. On the monitoring equipment, the scientists could hear the distinct pop of a neuron firing every time the rat entered a specific quadrant of the circle. No matter how many times the rat left that area and returned, the same specific cell would reactivate with perfect reliability. It became clear that the brain was not just reacting to the world, but was actively constructing a stable internal model of it. These cells are so dedicated to their specific locations that they form the very foundation of our spatial memory. They allow us to navigate through our homes with an effortless grace that we often take for granted. This internal mapping system is constantly updating, yet it possesses a quiet stability that keeps our world from feeling chaotic or fragmented. It is a silent, rhythmic process that happens beneath the surface of our conscious thought. Interestingly, this mapping process does not rely solely on what we can see with our eyes. Even when the lights are extinguished and the world is draped in shadow, these cells continue their vigilant work, maintaining our orientation in ways that seem to defy the limits of our five senses. The human brain possesses an extraordinary ability to reorganize its internal architecture the moment you step across a threshold into a new environment. This biological phenomenon is known as remapping, a process where the hippocampus completely reshuffles its neural firing patterns to create a fresh template for a new space. When you leave your living room and enter your kitchen, the specific group of place cells that were active moments ago suddenly falls silent, replaced by an entirely different ensemble of neurons. It is as if the brain is swapping out one physical map for another, ensuring that the coordinates for the sofa never become tangled with the coordinates for the stove. This transition happens almost instantaneously, allowing for a seamless transition between different spheres of our daily lives. This system is so sophisticated that it can distinguish between two rooms that are visually identical in every way. Imagine two circular rooms with the same white walls, the same lighting, and the same floor texture, separated only by a short hallway. Even if the sensory input is exactly the same, the brain uses subtle contextual clues like the direction of the entry point or the relative distance from a previous landmark to tell them apart. Researchers have observed that the hippocampal map will flip its orientation or use a completely different set of cells for the second room to prevent a navigational error. This prevents the mind from becoming lost in a repetitive or mirrored environment, providing a unique identity to every location we visit. The stability of these neural maps is perhaps one of the most comforting aspects of our biological programming. Once a map is created and stored, it possesses a remarkable degree of permanence that can last for years without being accessed. You might not visit a specific park or a friend's childhood home for an entire decade, yet the moment you pull into the driveway, the original neural map reactivates with startling precision. According to foundational texts like Campbell Biology, these spatial firing rates are among the most stable rhythmic patterns in the entire nervous system. This suggests that our sense of place is not a fleeting thought, but a physical structure etched into the very fabric of our temporal lobes. Have you ever wondered why walking into your childhood bedroom can instantly trigger a flood of specific, localized memories that you haven't thought about in years? This instant recognition occurs because the place cells associated with that specific room have remained in a dormant but preserved state, waiting for the correct environmental trigger to fire once again. The brain does not need to relearn the geometry of the walls or the distance from the bed to the door because the original blueprint was never deleted. Instead, the hippocampus simply reloads the archived file, bringing with it all the emotional and sensory data that was originally attached to those coordinates. This creates a powerful sense of continuity in our lives, anchoring our personal history to the physical world in a way that feels both solid and reliable. The persistence of these maps is a testament to the brain's efficiency in managing massive amounts of spatial data without constant upkeep. While other types of memories might fade or become distorted over time, the geometric relationship between landmarks in a well-known space tends to remain vivid and accurate. This spatial integrity is what allows a person to navigate their home in total darkness or find their way back to a favorite hiking trail after a long absence. The brain treats space as the ultimate filing system, using the physical layout of the world as a skeletal structure upon which all other experiences are hung. As we move through the world, these maps are constantly being refined and reinforced by our movements and our interactions. Every time we walk a familiar route, the connections between the specific place cells involved in that journey become slightly stronger and more efficient. This silent background process ensures that we never truly lose our way in the places that matter most to us. It is a quiet form of biological insurance, guaranteeing that no matter how much time passes, the internal compass of the mind will always know how to find its way back to a familiar shore. This stability provides the foundation for everything we understand about our position in the universe. Deep within the temporal lobe lies a sophisticated gateway known as the entorhinal cortex. This region serves as a vital bridge, connecting the memory-rich hippocampus to the vast analytical power of the neocortex. While the hippocampus excels at creating specific snapshots of an event, it relies heavily on the entorhinal cortex to provide a reliable coordinate system for those memories. Without this constant stream of spatial data, our internal maps would lack a mathematical foundation. This brain structure acts as the primary sensory interface, filtering information from the outside world before it ever reaches the deep memory centers. It sits in a strategically perfect location to gather data from the eyes, the ears, and even the skin, translating those signals into a language the rest of the brain can understand. Scientists often describe this area as the brain's high-speed switchboard. Every piece of information about where we are standing is processed here first, ensuring that the spatial where is always attached to the experiential what. The entorhinal cortex does not just pass information along. It actually generates the raw geometric data required for navigation. It functions much like the underlying grid on a piece of graph paper, providing the literal X and Y coordinates that the hippocampus then uses to plot specific landmarks. If you imagine the hippocampus as an artist painting a landscape, the entorhinal cortex is the person who measured the canvas and drew the perspective lines first. This relationship ensures that our memories are not just floating fragments but are instead pinned to a stable, predictable physical reality. Because of its role as a master coordinator, this region is essential for the way we integrate new experiences into our long-term understanding of the world. It allows the brain to compare a current location with every other place a person has ever visited. This constant comparison happens below the level of conscious thought, providing a seamless sense of continuity as we move from one room to another. By maintaining this flow of data, The entorhinal cortex helps create a unified sense of space that persists even when we are not actively thinking about our surroundings. It is the silent architect of our internal world, working tirelessly to keep our mental maps aligned with the physical truth of the environment. While the hippocampus identifies specific locations, another remarkable discovery revealed how the brain maintains a universal sense of scale and distance. In the early 2000s, researchers Maybrit and Edvard Moser identified a specific type of neuron in the entorhinal cortex that functions quite differently from a place cell. These neurons, now famously known as grid cells, do not fire in just one spot within an environment. Instead, a single grid cell will activate at multiple, regularly spaced locations that form a strikingly precise geometric pattern. When an organism moves through any given space, these cells fire in a way that maps out the floor into a series of equilateral triangles. If you were to connect the dots of every location where one of these cells activates, you would see a perfect hexagonal tiling covering the entire room. This rigid, repeating lattice is one of the most mathematically structured phenomena ever observed within a living brain. According to research published in the journal Nature, this discovery was so fundamental to our understanding of the mind that it earned the Mosers a Nobel Prize in Physiology or Medicine. This internal hexagonal lattice serves as a biological coordinate system, functioning much like the latitude and longitude lines on a physical map. Unlike a landmark that tells you where a specific building is, a grid system provides a consistent way to measure the distance between any 2 points. Even if you are standing in a completely featureless desert or a dark room with no visual cues, your grid cells provide a steady, reliable metric for how far you have traveled. This allows the brain to calculate its position relative to a starting point using only the internal geometry of the neural map. The beauty of this system lies in its incredible regularity and its independence from the external world. While a place cell might change its behavior based on the color of the walls or the furniture in a room, grid cells maintain their hexagonal spacing regardless of the environment's appearance. This suggests that the brain possesses an innate, preconfigured sense of two-dimensional space that it can project onto any new location. It is as if the mind carries a translucent sheet of graph paper that it overlays onto the world to ensure every step is accounted for with mathematical precision. By providing this stable geometric framework, grid cells allow the brain to perform complex spatial calculations without needing to see where it is going. This process, often compared to the way a modern GPS calculates coordinates, ensures that the internal map remains proportional to the physical world. The hexagonal arrangement is particularly efficient because it provides the highest resolution for spatial coverage with the fewest possible firing points. This natural optimization allows the brain to track movement across vast distances while using a relatively small number of neurons to maintain the integrity of the map. Because these cells are organized in different scales, some mapping small areas and others covering larger spans, the brain can track movement with both fine detail and broad perspective simultaneously. This layered approach ensures that whether you are reaching for a cup on a table or walking across a city park, your brain has a dedicated geometric layer to handle the specific scale of the task. This constant, rhythmic firing of grid cells provides the silent foundation for every move we make, turning the abstract concept of space into a tangible, navigable reality. Beyond the hexagonal lattices of the grid system, the brain possesses a specialized group of neurons known as head direction cells that function as an internal compass. Unlike the place cells which tell you where you are, these unique neurons are dedicated solely to informing you which way you are currently facing. These cells operate with a remarkable level of independence, firing based on the orientation of your head regardless of your physical location or the posture of your body. A head direction cell will remain active whether you are standing in the kitchen or walking through a park, provided your head is pointed in its preferred cardinal direction. This system effectively decouples your sense of orientation from your sense of place, allowing the brain to track two different streams of spatial data simultaneously. Information from these neurons flows directly into the navigation circuits of the hippocampus, providing a constant directional anchor for the maps being built there. It is a silent, internal orientation that remains steady even when the external world provides no visual landmarks to guide your path. The primary source of information for these cells comes from the vestibular system located deep within the structures of your inner ear. As you turn your head, the fluid in your semicircular canals shifts, sending immediate signals to the brain about the speed and angle of the rotation. This physical movement is translated into neural activity, ensuring that your internal compass is updated in real time as you navigate through a room. The brain integrates these vestibular signals with motor commands, creating a seamless loop that tracks every slight tilt or turn of the head. Because this system relies on internal mechanical sensors, it functions perfectly in total darkness, allowing you to maintain a sense of direction without sight. You can experience the quiet efficiency of these cells during a typical night of bedtime listening when you are resting in a dark room. If you were to close your eyes and slowly rotate your head, you would still possess an intuitive understanding of where the door or the window is located. Even without a single photon of light reaching your retinas, your head direction cells are firing rhythmically to keep your internal map aligned with the room. This constant awareness of your orientation is one of the many relaxing facts that demonstrate how the brain maintains your safety and stability automatically. In the context of science for sleep, understanding this system helps illustrate why we feel a sense of groundedness when we settle into a familiar environment. Research published in the journal Nature suggests that these cells can even maintain their orientation during sleep, though their activity patterns shift during different stages of rest. This internal compass ensures that when you wake up in the middle of the night, your brain can instantly recalibrate your position within the space around you. The elegance of the head direction system lies in its ability to provide a stable reference point in an ever-changing and often unpredictable world. The internal map within your brain requires more than just a sense of direction and a grid of coordinates to function effectively. It needs a way to define the limits of the world around you so that your movements remain grounded in physical reality. This essential task is handled by a specialized class of neurons known as border cells, which were first identified in the medial entorhinal cortex. These cells serve as the biological equivalent of a perimeter fence, providing the brain with a constant readout of where a space begins and where it ends. Without these specialized sensors, your mental map would have no edges, making it nearly impossible to navigate a room without constantly colliding with the environment. Border cells operate with remarkable specificity by firing only when you are in close proximity to a physical boundary. Unlike place cells, which might fire in the center of a room, a border cell remains silent until you approach a wall, a steep drop-off, or even the edge of a table. As you walk toward the perimeter of a space, these neurons begin to pulse with activity, signaling to the rest of the navigational system that a limit has been reached. This neural feedback allows you to walk through a dark hallway with confidence, as your brain instinctively calculates the distance between your body and the solid surfaces nearby. The activity of these cells is not merely a warning system but a fundamental anchoring mechanism for the entire cognitive map. When you enter a new environment, the brain immediately looks for geometric markers like corners and long straight edges to establish a frame of reference. By identifying these boundaries, the hippocampus can lock its grid and place cell patterns into a fixed position. This process ensures that your internal map does not drift or rotate incorrectly while you move. The geometry of a room acts as a skeletal structure upon which all other spatial memories are draped, providing a stable foundation for your sense of place. Scientists have observed fascinating behaviors in these cells when the physical environment is manipulated in a laboratory setting. If a wall in a familiar room is suddenly moved several feet outward, the border cells will often stretch their firing fields or shift their activity to match the new perimeter. This demonstrates that the brain is constantly updating its understanding of environmental limits based on real-time sensory input. If a new partition is dropped into the middle of an open space, a new set of border cells will quickly recruit themselves to define this fresh boundary. This flexibility is what allows you to adapt to a world that is frequently changing, from rearranged furniture to the construction of new buildings. These cells are particularly important for maintaining safety and preventing accidents in high-stakes environments. When a mountaineer stands near the edge of a cliff, their border cells are firing at an intense rate, providing the brain with the precise spatial data needed to maintain a safe distance from the ledge. Even in a familiar home, these neurons are quietly working as you navigate around a kitchen island or step through a doorway. They provide a sense of containment and security, ensuring that your mental representation of the world matches the physical obstacles you encounter. The presence of these boundary-sensing neurons raises an intriguing question about how the brain handles environments that lack any clear physical limits. While a room or a forest has distinct edges to anchor a map, some spaces seem to stretch on forever into a vast, featureless horizon. One might wonder how the hippocampal system maintains its accuracy when an organism transitions into an infinite, boundaryless expanse like the open ocean or the endless blue of a clear sky. Exploring how the mind adapts to these limitless voids reveals even more about the hidden depths of our biological GPS. The internal navigation system requires more than just a map of static boundaries and specific locations. To remain truly oriented, the brain must also understand the physics of its own movement through the world. This is where a specialized class of neurons known as speed cells comes into play, serving as the biological speedometer for the mind. These cells were identified by researchers at the Kavli Institute for Systems Neuroscience, who discovered that certain neurons in the entorhinal cortex are dedicated solely to tracking how fast an organism is traveling. Unlike place cells, which care about where you are, or head direction cells, which care about where you are facing, speed cells focus entirely on the velocity of your motion. The behavior of these cells is remarkably precise and mathematically consistent. As you begin to move, these neurons start to fire in a rhythmic pattern that correlates directly with your physical pace. If you transition from a slow, leisurely stroll to a brisk walk, the firing rate of these speed cells increases in a linear, proportional fashion. Should you break into a full run, the electrical signals from these neurons intensify even further, providing the rest of the hippocampal formation with a real-time data stream of your current velocity. This information is broadcasted regardless of the direction you are heading or the specific landmarks you might be passing at that moment. By maintaining this constant tally of velocity, the brain performs a sophisticated calculation known as path integration. It essentially multiplies the speed provided by these cells by the duration of the movement to determine exactly how far you have traveled from a known starting point. This allows the internal map to update your position even when your eyes are closed or when you are moving through a featureless environment. It is a seamless, subconscious process that ensures your mental representation of space stays perfectly synchronized with your physical body. Consider the simple experience of waking up in the middle of the night and needing to reach a light switch on the opposite side of a pitch-black room. Even without any visual cues to guide you, you can intuitively feel how many steps are required to cross the floor. As you move at a steady, cautious pace, your speed cells are firing at a consistent rate, telling your brain exactly how much distance you are covering with every passing second. You reach out your hand at precisely the right moment because your internal GPS has tracked your velocity and updated your mental coordinates with every inch of progress. This elegant biological mechanism allows you to navigate the darkness with a quiet, grounded confidence in your own position. Path integration represents one of the most sophisticated computational feats performed by the brain, often referred to in classical navigation as dead reckoning. This internal process allows a living being to track its current position and orientation by purely monitoring its own movement away from a known starting point. Without the need for any external landmarks, the mind creates a continuous vector that points back to home, allowing for a direct return even after a winding or complex journey. This ability is found across the animal kingdom, from desert ants navigating featureless salt pans to humans walking through a dark house at night. This remarkable system relies heavily on self-motion signals known collectively as proprioception, which is the body's internal sense of its own position in space. As you move, your brain receives constant updates from your muscles, joints, and the vestibular system in your inner ear. These sensors detect every change in acceleration, every rotation of the neck, and every step taken by the legs. By integrating these physical sensations over time, the brain calculates the precise distance and direction of the displacement, essentially drawing an invisible line through the environment. When you walk in a straight line with your eyes closed, you are using this internal geometry to estimate your progress. The brain is not guessing. It is performing a high-speed mathematical integration of your velocity and your heading. If you turn a corner, your head direction cells signal the change in orientation, and your speed cells update the rate of travel. Together, they allow you to maintain an intuitive sense of where your starting point is located, even if it is now behind a wall or far out of sight. However, this internal system is not perfectly immune to the passage of time or the accumulation of small physical discrepancies. In the absence of visual feedback, minor errors in the perception of a turn or the length of a stride begin to compound. Research published in journals like Science has shown that when humans are asked to walk in a straight line through a vast, featureless desert or a dense forest without a compass, they eventually begin to walk in circles. Without the reset button provided by a visible landmark, the internal map slowly drifts away from physical reality. Despite these eventual limits, path integration remains a beautifully calming and automatic background process that happens without any conscious effort. You do not have to think about the trigonometry of your movements or consciously calculate the angles of your turns. Your hippocampus handles this complex data stream in the silence of the subconscious, keeping you anchored to the world around you. It provides a quiet sense of stability, ensuring that you are never truly lost as long as your internal compass remains active. A gentle shift in focus brings us from these hidden cellular mechanics toward the broader ways we behave within our mental landscapes. The study of how we use these internal maps has led to the development of the cognitive map theory in behavioral psychology. This theory suggests that we do not just react to individual stimuli in our environment, but instead build a comprehensive, holistic representation of the world. Even when we are not looking for a specific destination, our brains are passively absorbing the layout of the rooms we inhabit and the streets we frequent. This latent learning ensures that when a need arises, the map is already waiting, fully formed and ready to guide us through the geometry of our lives. Edward Tolman, a pioneering psychologist in the mid-20th century, challenged the prevailing ideas of his time by proposing that organisms do more than just react to their surroundings. Before his work, many scientists believed that learning was merely a series of reinforced habits, where a specific action was triggered by a specific reward. Tolman suspected that something far more sophisticated was happening inside the mind, suggesting that animals develop what he called a cognitive map. This internal representation is a mental model of the environment that allows for flexible navigation rather than just a robotic following of paths. To prove this, Tolman conducted a series of elegant behavioral experiments involving rats in complex wooden mazes. In one famous study, he allowed a group of rats to wander through a maze for several days without providing any food or reward at the end. To a casual observer, these rats appeared to be learning nothing at all, as they meandered aimlessly through the various corridors. However, as soon as food was placed in a specific goal box, these previously unrewarded rats found the target just as quickly as rats that had been trained with rewards from the very beginning. This discovery led to the formal definition of latent learning, which describes the brain's ability to process and store spatial information without any immediate incentive. It suggests that our minds are constantly and passively absorbing the layout of the world, even when we have no specific destination in mind. You might experience this when you walk through a new park or a quiet library for the first time. Though you aren't looking for anything in particular, your brain is silently tiling the floor plan into your consciousness. This background mapping ensures that if you ever need to find an exit or a specific shelf later, the information is already waiting for you. Tolman's research demonstrated that these cognitive maps allow for a high degree of spatial problem solving, such as taking a shortcut when a primary path is blocked. This ability proved that the rats weren't just memorizing a sequence of left and right turns, but were instead consulting a mental overhead view of the entire space. It was a revolutionary shift in psychology, moving the focus from simple outward behavior to the complex internal world of mental representation. This internal map is a foundational piece of our biology that provides a sense of continuity and predictability to our daily lives. These mental maps eventually serve a purpose that extends far beyond just finding our way from one room to another. Modern neuroscience, including research found in foundational texts like Campbell Biology, suggests that this spatial scaffolding is the literal foundation for human episodic memory. Because the hippocampus maps out where an event happens, it provides the stage upon which the rest of the memory is built. Every story you remember about your life is anchored to a specific coordinate on your internal map, linking the geometry of the world to the narrative of your personal history. This deep connection between space and memory is why walking into a childhood home can suddenly trigger a flood of specific, long-forgotten lived experiences. The quiet, rhythmic pulse of slow-wave sleep marks the beginning of a profound architectural project within the human brain. While the body remains nearly motionless, the hippocampus enters a state of intense, focused activity known as hippocampal replay. During these deep stages of rest, the brain begins to systematically revisit the paths traveled and the spaces explored during the preceding hours of wakefulness. This is not a random firing of neurons, but a highly organized, chronological reenactment of the day's spatial experiences. Every street turned, every hallway walked, and every room entered is brought back to the foreground of neural activity in a silent, internal performance. This replay phenomenon is characterized by an incredible increase in processing speed compared to real-time experience. When you are awake and moving through a physical environment, your place cells fire in a specific sequence that mirrors your actual pace. However, once you fall asleep, the hippocampus compresses these exact sequences into high-speed bursts that occur up to 20 times faster than the original event. A 10-minute walk through a neighborhood can be replayed by the brain in a fraction of a second, allowing for the rapid-fire reinforcement of spatial coordinates. This compression allows the brain to iterate through multiple paths and experiences thousands of times in a single night of rest. The mechanism behind this high-speed data transfer involves a specific electrical pattern called sharp wave ripples. These ripples are among the most synchronous and powerful electrical events in the mammalian central nervous system, originating in the hippocampus and radiating outward. They act as a biological bridge, carrying the refined spatial data from the temporary storage of the hippocampus to the permanent archives of the neocortex. By firing in these intense, concentrated bursts, the brain ensures that the map of your world is etched into more stable neural structures. This process effectively offloads the day's cartography into long-term memory, freeing up the hippocampus to map new environments the following day. Science for sleep reveals that this nightly processing is essential for the stabilization of our mental maps. Without this coordinated replay, the subtle details of a new environment would likely fade into a blur of disconnected sensory impressions. Instead, the brain uses the stillness of the night to carefully stitch together the geometry of our lives, ensuring that our sense of place remains coherent and reliable. It is a form of internal maintenance that happens entirely beneath the level of conscious awareness, transforming a day of movement into a lifetime of orientation. This constant dialogue between different brain regions turns fleeting glimpses of a landscape into a permanent mental landmark. Could it be that the vivid, often fragmented landscapes we encounter in our dreams are simply the byproduct of this massive spatial data sorting process? If the brain is busy compressing, replaying, and filing away the coordinates of our physical world, it stands to reason that our internal vision might catch glimpses of these shifting maps. While the purpose of dreaming remains a subject of intense scientific study, the physical reality of spatial replay is well documented in modern neuroscience. The brain seems to prioritize the integrity of its inner GPS above almost all other informational tasks during the deep hours of the night. The physical wiring of these maps into our long-term memory is what allows us to wake up in a familiar room and immediately know where the door is located. This immediate, intuitive grasp of our surroundings is the direct result of the sharp wave ripples that occurred while we were dead to the world. Each night of rest acts as a reinforcement layer, thickening the neural pathways that represent our most frequented locations. Through this tireless repetition, the brain builds a world that feels solid, predictable, and safe. Learning about these relaxing facts helps us appreciate the sophisticated work our minds perform to keep us grounded in reality. This nightly consolidation is also a key reason why sleep deprivation leads to such profound spatial and navigational confusion. When the brain is denied the opportunity to perform its replay cycles, the spatial data gathered during the day remains fragile and unorganized. The sharp wave ripples never get the chance to broadcast their coordinates to the cortex, and the internal map begins to fray at the edges. Research published in journals like Nature and Science underscores that a single night of lost sleep can significantly impair a person's ability to remember a new route. The brain requires that window of quiet to turn a path into a memory. As we rest, the hippocampus isn't just looking backward. It is also preparing the foundation for every movement we will make tomorrow. By solidifying the maps of the past, it provides the stable ground upon which we plan our future trajectories. This invisible cartography is a testament to the brain's dedication to spatial order. Even in the deepest darkness, the mind is busy illuminating the paths we have taken, ensuring we never truly lose our way in the world we call home. The map is always being updated, always being refined, and always being saved for the journey ahead. While the brain is famous for replaying the past during rest, it possesses an even more mysterious ability known as preplay. This phenomenon occurs when the neurons in the hippocampus fire in a sequence that describes a path the individual has not yet taken but is likely to follow soon. It is as if the internal GPS is running a high-speed simulation of a future journey before the first step is even made. This predictive firing happens in the blink of an eye, allowing the mind to preview a route with startling mechanical precision. Scientific observations of these neural sequences show that the brain actually explores potential paths at a rate much faster than physical movement allows. When a person pauses at a crossroads or considers a new direction, the hippocampus scans the available options by firing cells in a linear chain toward the intended goal. This quiet internal rehearsal serves as a biological dry run for reality. By simulating the walk to a destination, the brain can pre-calculate the effort and distance required for the trip. This ancient system for navigating physical landscapes eventually evolved into our sophisticated ability to navigate abstract choices. The same neural architecture used by early ancestors to find water or avoid predators is now used by the modern mind to weigh complex life decisions. We imagine different futures by literally walking through them in our minds, using the spatial map as a canvas for our intentions. This evolutionary pivot suggests that human foresight is deeply rooted in our mechanical need to move through three-dimensional space. The brain performs these thousands of hypothetical simulations with a calm, silent efficiency that we rarely notice in our daily lives. Within milliseconds, the mind can test out a dozen different variations of a single plan, discarding the ones that lead to dead ends. This process happens in a low-stimulation background state, often when we are daydreaming or sitting in quiet reflection. It is a testament to the brain's power that it can build entire alternate realities without consuming vast amounts of conscious energy. It is a profound realization of neuroscience that our spatial hardware and our imagination hardware are essentially the same thing. To remember where we have been, to know where we are, and to imagine where we might go all require the same hippocampal map. Our ability to dream of a better future is a direct gift from the cells that originally helped us find our way home in the dark. This elegant internal geometry ensures that we are never truly lost, even when we are exploring the uncharted territories of our own thoughts. Human biology links our sense of place to our personal history through a deep structural connection in the temporal lobe. This relationship exists because the hippocampus serves a dual purpose as both a spatial navigator and a recorder of life events. When we experience something new, The brain does not simply store a flat image or a string of words. Instead, it embeds that information directly into a three-dimensional coordinate system that represents our physical environment. This biological reality ensures that every memory we possess is anchored to a specific location in the world. Our minds often prioritize the where of an event long before we can accurately recall the when. You might struggle to remember the exact Tuesday or Thursday a specific conversation occurred, yet you can likely visualize the exact street corner or kitchen table where you stood. This happens because the brain treats space as the primary scaffolding for all episodic data. Before a memory is filed away, it is stamped with a spatial signature that makes the location the most accessible retrieval queue in our mental filing cabinet. This inextricable link is why spatial disorientation and memory loss frequently emerge as simultaneous symptoms in clinical settings. Major neuroscience texts, such as Candel's Principles of Neuroscience, detail how damage to the hippocampal formation disrupts the ability to form new memories while also destroying the internal map. When the cellular architecture that builds our sense of here begins to falter, the ability to sequence the what of our lives inevitably follows. A person may find themselves unable to navigate a familiar neighborhood because the same neurons required for direction are also responsible for holding on to the recent past. Consider the common experience of walking into a room only to realize you have completely forgotten your purpose for being there. This momentary lapse occurs because the brain has performed a remapping process as you cross the threshold into a new environment. Your previous intention was physically and neurologically tied to the room you just left. The spatial context of the hallway or the kitchen held the memory of your goal, and entering a new space caused the brain to reset its active map. To recover that lost thought, you might naturally find yourself walking back to the original room where the idea first formed. As you cross back over the threshold, the familiar geometry of the previous space reactivates the specific cluster of place cells associated with your intent. The memory often snaps back into focus the moment your body returns to the correct coordinates. This simple, everyday occurrence illustrates how our thoughts are not floating freely in a vacuum but are tethered to the physical world around us. Our internal GPS does more than just prevent us from getting lost in the woods or on a city street. It provides the literal ground upon which our entire identity is built, allowing us to weave a continuous story of who we are. By anchoring our experiences to the stable features of the landscape, the brain ensures that our history remains organized and retrievable. This quiet, constant partnership between space and memory allows us to move through the world with a profound sense of continuity and belonging. The ancient Greeks and Romans developed a powerful mnemonic strategy known as the method of loci, or the memory palace, long before the invention of modern printing or digital storage. This technique relies on the brain's innate ability to remember spatial environments with far greater precision than abstract lists of words or numbers. By mentally placing pieces of information along a familiar path or within a specific room, a person can create a durable internal archive of knowledge. The term loci is the Latin word for places, reflecting the foundational idea that human memory is fundamentally tied to geographical coordinates. Ancient orators used this spatial strategy to memorize lengthy speeches, ensuring they never lost their place during hours of public delivery. They would mentally walk through a well-known building, such as their own home or a public forum, depositing specific concepts at distinct landmarks like a doorway, a statue, or a window. To retrieve the information, they simply retraced their steps in their mind's eye, seeing each concept waiting for them at its assigned location. This transformation of abstract data into a physical journey allowed for the effortless recall of thousands of items in a precise, unbreakable sequence. The effectiveness of the memory palace is not merely a psychological quirk, but is deeply rooted in the biological architecture of the human brain. When a person visualizes a familiar room, the grid cells in the entorhinal cortex and the place cells in the hippocampus activate just as they would during actual physical navigation. This process essentially hijacks the evolutionary hardware designed for survival and spatial orientation, repurposing it for the storage of complex, non-spatial information. By attaching a name or a date to a specific corner of a room, the brain treats that information as a vital landmark within a physical territory. Neuroscience research indicates that the hippocampus is exceptionally efficient at encoding spatial maps because, for most of evolutionary history, knowing the layout of a landscape was a matter of life or death. While the brain may struggle to memorize a list of 30 random objects, it can easily remember the layout of a childhood home where thousands of objects were once located. The method of loci takes advantage of this high-bandwidth spatial processing system to anchor fleeting thoughts into a stable, three-dimensional framework. This creates a much stronger neural trace than rote repetition or simple verbal memorization ever could. The structural stability of these mental palaces allows the information stored within them to remain accessible for years, even if it is not frequently reviewed. Because the grid cells provide a consistent geometric lattice, the brain can jump to any specific room in the memory palace and find the stored data exactly where it was left. This spatial anchoring prevents the interference that often causes different memories to blur together or fade over time. The information becomes part of the environment itself, benefiting from the same neural longevity as the memory of one's own neighborhood. Observing how the brain handles these imagined spaces reveals that the mind does not distinguish between a physical building and a vividly imagined one when it comes to memory encoding. Modern brain imaging shows that the same navigational circuits fire with intense synchronization during the use of the method of loci. This bridge between the physical world and the world of thought demonstrates how deeply our cognitive abilities are tied to the simple act of moving through space. The memory palace remains one of the most striking examples of how biological evolution continues to shape the way humans process the complexities of the modern world. One of the most famous examples of the brain's ability to physically reshape itself comes from a landmark study involving the streets of London. To become a licensed taxi driver in that sprawling city, applicants must master an incredibly difficult test known as the knowledge. This process typically takes 3 to 4 years of intense daily study, as drivers must memorize the layout of 25,000 individual streets. They also have to learn the locations of thousands of landmarks, parks, and small businesses tucked away in the city's labyrinthine center. This grueling mental challenge requires the brain to build a massive, high-resolution internal map of one of the world's most complex urban environments. Researchers at University College London decided to investigate whether this monumental task left a physical mark on the driver's brains. They used structural MRI scans to look deep into the temporal lobes of these experienced navigators. What they discovered changed the way the scientific community thinks about adult neuroplasticity and the capacity for the brain to grow. The study revealed that the posterior hippocampus, the specific region of the brain responsible for spatial navigation, was significantly larger in the taxi drivers than in the general population. This was not a subtle difference. The volume of gray matter in this area had expanded to accommodate the sheer amount of spatial data required to navigate London without a map. The researchers found that the more years a driver had spent on the job, the larger this part of their brain had become. To ensure these results were accurate, the scientists compared the taxi drivers to a control group of London bus drivers. The bus drivers lived in the same city and spent just as many hours behind the wheel every day, but their brains did not show the same structural growth. The critical difference was that the bus drivers followed the same fixed routes every shift and did not have to actively calculate new paths. Their brains were not required to constantly update a complex mental map, so the hippocampus did not receive the same intensive workout. This comparison proved that it was the active, effortful navigation, not just the act of driving, that caused the physical expansion of the brain. The hippocampus acts much like a muscle in this regard, growing stronger and more dense when it is pushed to its limits by a challenging environment. This study was a breakthrough because it showed that even in adulthood, the brain is not a static organ. It remains dynamic and capable of structural change well into middle age and beyond. The physical changes were so specific that the researchers could almost see the map of London reflected in the density of the neurons. The posterior hippocampus seems to act as a storage vault for these established spatial representations, holding the coordinates for every turn and alleyway. As the drivers successfully passed the knowledge, their brains literally carved out more room for the city. This reinforces the idea that our internal maps are living structures that respond to how we choose to use them. However, the brain is also incredibly efficient, and it rarely maintains tissue that it no longer perceives as necessary for survival or daily function. In an era where we increasingly rely on external devices to find our way, scientists have begun to wonder what happens when we stop exercising this internal muscle. There is a growing body of evidence suggesting that delegating our navigation to a screen might have unintended consequences for the very architecture of the mind. This raises a quiet but persistent question about the long-term health of our spatial memory in a world dominated by satellite guidance. If the brain can grow to accommodate 25,000 streets, it might also be capable of shrinking when those streets are no longer stored within. The delicate balance between our biological maps and our digital tools is a subject of intense modern study. Understanding how these changes occur helps us appreciate the incredible work the brain does every time we find our way home. The origins of our internal mapping system are deeply rooted in the earliest chapters of biological history. Before there were complex thoughts or spoken languages, there was simply the necessity of movement. For any organism to survive, it had to solve the fundamental problem of where to go and how to get back. This basic requirement for spatial awareness acted as a primary driver for the evolution of the modern brain. In the silent depths of ancient oceans, the transition from a passive existence to an active one changed everything for life on Earth. Early life forms were often stationary, acting as filter feeders that waited for nutrients to drift toward them on the current. These organisms had very little need for a centralized nervous system because their environment came to them. However, as some species began to move intentionally to forage for food, the biological stakes were raised immediately. This shift toward active foraging required a way to track the location of resources and the presence of predators. A brain that could not remember where it had already been was a brain that would likely starve or be eaten. Movement through a physical environment is never a simple act of repositioning a body. It involves a continuous and sophisticated process of risk assessment that must happen in real time. An animal moving through a forest or a reef must constantly evaluate the safety of its path while planning the most efficient route to its goal. This creates a massive computational load on the nervous system, requiring the integration of sensory data with internal memories. Every successful journey reinforces the neural pathways that represent a safe passage through the world. To navigate effectively, an organism must also have a secure place to retreat when its energy is depleted. The ability to find a hidden burrow or a high branch is what allows a creature to eventually fall asleep without being vulnerable to the many dangers of the wild. This sense of spatial security is perhaps the most ancient form of comfort we know. Our ancestors relied on their internal maps to guide them back to the safety of the hearth or the cave. Even today, the quiet confidence of knowing exactly where we are in space is a biological signal that it is finally safe to rest. The biological world offers a strange and revealing story in the life of a creature called the sea squirt or tunicate. This small marine invertebrate begins its life in a form that looks very much like a microscopic tadpole. During this larval stage, the sea squirt is a modal traveler, swimming through the vast currents of the ocean in search of a permanent home. To accomplish this task, it possesses a rudimentary but functional nervous system, including a simple brain-like structure known as a cerebral ganglion. This tiny command center is essential because the larva must navigate complex underwater environments, sensing light and the pull of gravity to find the perfect spot to settle. As it swims, the larval sea squirt uses its chordate features, like a stiffening rod called a notochord, to propel itself toward a suitable substrate. It is looking for a specific kind of rock or a stable piece of coral where it can anchor itself for the remainder of its existence. During this period of its life, the sea squirt is an active participant in the spatial world, constantly processing sensory data to avoid obstacles and identify a safe harbor. Its brain is its most valuable tool, allowing it to interpret the three-dimensional geometry of the seafloor. Without this internal mapping ability, the young organism would simply drift aimlessly, likely failing to find the resources it needs to survive. Once the sea squirt finds its ideal location, it undergoes one of the most remarkable transformations in all of biology. It attaches its head to the chosen surface and begins a process of radical metamorphosis. Within a matter of hours, the creature begins to absorb its own tail and its specialized swimming hardware. Most strikingly, the sea squirt then proceeds to digest its own brain. Because it has transitioned from a mobile explorer to a stationary filter feeder, it no longer needs the complex neural machinery required for navigation. The animal effectively eats the very organ that allowed it to find its home in the first place. This biological pivot highlights a profound truth about the nature of intelligence and the purpose of a nervous system. Maintaining a brain is an incredibly expensive endeavor in terms of metabolic energy. In many species, the brain consumes a disproportionate amount of the body's total oxygen and nutrient supply. For the sea squirt, once the requirement for movement through space has ended, the energetic cost of keeping a navigational brain is no longer justifiable. The creature simply discards the faculty of thought because it no longer has any place to go. This suggests that the primary function of complex brains in the animal kingdom is not just to think, but specifically to coordinate movement through an unpredictable world. When an organism becomes a permanent part of the landscape, the need for a cognitive map vanishes entirely. The sea squirt becomes a living example of how deeply our mental lives are tied to our physical journeys. Without the need to navigate, the internal world of the creature falls silent. The quiet stillness of a stationary life marks the end of one kind of existence and the beginning of another. As we look beyond the simple life of the sea squirt, we find that more mobile animals have developed even more sophisticated ways to track their place in the world. The intricate dance between movement and memory is perhaps nowhere more evident than in the minds of birds that must survive long, frozen winters. In the high-altitude forests of the American West, a bird known as Clark's nutcracker performs a feat of spatial memory that seems almost impossible. During the late summer and autumn, a single nutcracker will collect and bury up to 30,000 pine seeds across an area covering many square miles. These seeds are not hidden in one central pantry but are distributed in thousands of small, distinct caches. Each hiding spot contains only a few seeds, tucked away under the soil or tucked into the bark of trees. The bird does not rely on smell to find these treasures later, as the ground is often covered by a thick, uniform blanket of snow. Instead, it relies on a highly developed internal map that stores the precise geometric coordinates of every single location it has used. This incredible ability is a cornerstone of avian neuroscience and demonstrates the specialized power of the bird's hippocampus. While a human might struggle to remember where they parked a car in a large lot, the nutcracker can relocate its caches with an accuracy rate of over 80% months after hiding them. It achieves this by triangulating the position of each cache relative to large, permanent landmarks like boulders or specific trees. The bird's brain must maintain a high-resolution 3D model of the entire landscape, accounting for how the scenery changes when viewed from different angles or under different weather conditions. This is not just a simple list of locations. It is a sophisticated, living document of a vast physical territory. The biological cost of maintaining such a massive database of information is reflected in the physical structure of the bird's brain. Research published in journals like Science and Nature has documented a fascinating phenomenon known as neuroplasticity in food caching birds. As the autumn approaches and the birds begin their frantic caching behavior, the volume of their hippocampus actually increases. New neurons are born and integrated into the existing spatial networks to accommodate the massive influx of new data. This seasonal expansion allows the brain to swell in capacity exactly when the demand for spatial memory is at its peak. It is a rare example of a vertebrate brain physically growing and shrinking in response to the environmental challenges of the seasons. Once the winter arrives and the need for new caching ends, the hippocampus may slowly decrease in size to conserve energy during the lean months. This elegant biological rhythm ensures that the bird is never carrying more neural weight than it absolutely needs to survive. You might think of this process as a library that builds new wings and shelves just in time for a massive delivery of rare books, only to close those sections once the information has been safely archived. The nutcracker's survival depends entirely on this accordion-like flexibility of the mind. It is a quiet, internal preparation that mirrors the stillness of the winter woods. There is a certain peace in imagining the bird's mind at work during a quiet winter night. Just as the seeds are tucked safely beneath the cold earth, the memories of those locations are filed away in the deep, rhythmic folds of the brain. Each coordinate is a promise of future sustenance, a tiny point of light on a mental map that guides the bird through the darkness and the cold. The act of remembering becomes a form of survival, a way to bridge the gap between the abundance of autumn and the scarcity of spring. This internal world is a sanctuary of information, held steady while the external world remains frozen and silent. Through the lens of science, we see that the mind is not just a tool for thinking, but a landscape where every memory is a seed waiting for the right moment to be found. Beyond the physical landmarks of mountains and rivers lies a silent, invisible grid that blankets the entire planet. This is the Earth's magnetic field, a shifting veil of energy generated by the churning liquid iron deep within the planetary core. While humans are largely blind to this force without the aid of a compass, many other species possess a biological sense known as magnetoreception. This allows them to perceive the north and south poles as tangible directions, turning the entire globe into a readable map. For a migrating bird or a sea turtle, the air and water are filled with directional data that remains constant even when the sun is hidden by thick clouds. Science has uncovered two primary theories for how animals actually see or feel these magnetic lines. One fascinating mechanism involves tiny crystals of magnetite, a naturally occurring magnetic mineral found in the tissues of some fish, birds, and even bees. These microscopic crystals act like internal compass needles, physically twisting or putting pressure on cell membranes as the animal moves through the magnetic field. This mechanical pull sends a direct signal to the brain, providing a constant readout of the animal's orientation relative to the poles. It is a primitive but incredibly reliable hardware system for staying on course across thousands of miles of featureless ocean. Another, even more mysterious mechanism involves a protein called cryptochrome, located within the retinas of certain migratory birds. This protein is sensitive to blue light and undergoes a quantum chemical reaction when exposed to the Earth's magnetic field. This reaction creates a radical pair of electrons that are highly sensitive to the angle of the magnetic lines. Some researchers believe this actually creates a visual overlay, allowing the bird to see the magnetic field as a pattern of light or shadow superimposed over their normal vision. In this way, the map of the world is literally painted onto the sky they fly through. This magnetic data provides more than just a simple north or south direction. It offers a sophisticated way to calculate latitude. The Earth's magnetic lines do not just run parallel to the surface. They curve and dip at different angles depending on how close you are to the poles. This is known as magnetic inclination. Near the equator, the lines are roughly parallel to the ground, but at the poles, they point almost straight down. By detecting this specific angle, a migrating animal can determine its exact distance from the equator, giving it a global coordinate system that never fades. Imagine for a moment if your own eyes could perceive these shimmering ribbons of energy stretching from horizon to horizon. How different would the world look if every street and forest was draped in a glowing, geometric lattice of magnetic force? This invisible architecture would make it impossible to ever truly feel lost, as the very air would hum with the signature of your location. For a creature with this gift, the planet isn't a chaotic wilderness, but a perfectly organized grid where every point has a unique magnetic fingerprint. When a migratory bird prepares for its journey, it overlays this magnetic inclination data onto its existing internal spatial map. The grid cells and place cells in its hippocampus likely integrate this invisible information to create a multidimensional understanding of space. This allows the bird to recognize not just where it is based on landmarks, but where it sits within the global magnetic field. It is a redundant system of navigation that ensures survival even when the weather turns and the stars are blotted out by a storm. This global grid acts as a stabilizing force for long-term spatial memory. While a forest might burn down or a coastline might erode over centuries, the magnetic field remains a relatively stable guidepost for generations. Young animals often inherit the ability to read these maps, embarking on their first migrations with a pre-programmed understanding of the magnetic highway. This inherited wisdom is a testament to the deep connection between the biology of the brain and the physics of the planet. As the sun sets and the visible world fades into shades of gray, the magnetic field remains as vibrant and clear as ever. For the nocturnal traveler, this silent guide is a constant companion, whispering the way home through the darkness. The Earth itself provides the map, and the brain has spent millions of years evolving the perfect sensors to read it. In the quiet of the night, this invisible dialogue between the planet and the mind continues, ensuring that every journey, no matter how long, has a destination. While many of the creatures we have discussed navigate on a mostly two-dimensional plane, the world changes entirely when an organism leaves the ground. For a bat soaring through a dense forest or a sprawling cavern, space is not a flat surface, but a vast, volumetric void. This three-dimensional environment presents a unique computational challenge for the brain, requiring it to track movement along the vertical axis with the same precision it uses for the horizontal. Navigating in this way means the brain cannot rely on a simple flat grid to understand its surroundings. Instead, it must construct a mental representation that accounts for height, depth, and the complex tilt of the body in midair. In the quiet darkness of the night, these nocturnal mammals move with a grace that suggests their internal maps are far more sophisticated than our own terrestrial versions. Researchers studying these remarkable animals eventually discovered the existence of specialized 3D place cells within the bat hippocampus. Unlike the place cells found in rats or humans, which typically fire based on a circular area on a floor, these cells fire when the bat enters a specific three-dimensional volume of air. It is as if the brain has divided the empty space of a room into a series of invisible, stacked boxes, each one monitored by a dedicated neuron. This discovery fundamentally changed our understanding of how the inner GPS functions in 3 dimensions. To complement these place cells, the bat's brain also utilizes 3D head direction cells. These neurons do not just track whether the animal is facing north or south, but also its pitch, roll, and yaw. This allows the bat to maintain a perfect sense of orientation even while performing acrobatic maneuvers or flying upside down. By integrating this directional data, the brain creates a spherical mental map rather than a flat cognitive one. This internal sphere allows the bat to perceive its environment as a continuous, wraparound reality where every coordinate is defined by its relation to the center. As the bat glides silently through the trees, its brain is constantly updating this spherical map, ensuring that every twist and turn is perfectly recorded. This seamless integration of height and direction represents one of the most elegant solutions to the problem of spatial navigation found in nature. The concept of a map usually implies a physical surface, yet the brain does not treat space as a static picture. Within the same hippocampal regions that track our physical coordinates, scientists have discovered a specialized class of neurons known as time cells. These remarkable cells fire in a predictable, rhythmic sequence to mark the passage of moments, even when a person or animal is standing perfectly still. They act as a neural metronome, providing a steady internal pulse that allows the mind to organize experiences into a linear narrative. This discovery reveals that our internal GPS is actually a 4-dimensional system, Space and time are not handled by separate departments of the brain, but are instead fundamentally mapped by the same underlying neural architecture. When you move through a landscape, the hippocampus is simultaneously tracking where you are and how long it has taken you to get there. This integration ensures that every memory we form is rooted in a specific context of both location and duration. To the brain, a journey is a single, unified ribbon of information where distance and time are inextricably linked. This connection is why we often measure the distance to a destination in minutes or hours rather than miles. The brain calculates the effort of travel by linking the velocity of our movement with the steady ticking of these internal time cells. As we navigate our world, this biological clock ensures that the sequence of events remains orderly and coherent. Consider the experience of sitting on a long-distance train as it moves steadily across a quiet, rural landscape. You might watch the horizon for what feels like an hour, and in your mind, that hour is physically tied to the specific miles of forest or field that rolled past your window. The time spent and the ground covered become a single unit of memory. Your brain does not store the trees and the minutes in separate folders. It weaves them together into a continuous tapestry of the journey. This seamless blending of dimensions allows us to revisit our past with incredible clarity. When you remember a specific walk through a park, your brain reactivates the place cells for the path and the time cells for the duration of the stroll. Because these neurons work in such close harmony, the memory feels like a reliving of the event rather than a static photograph. The same hardware that prevents us from getting lost in a forest also prevents us from getting lost in the timeline of our own lives. By anchoring time to space, the hippocampus creates a stable framework for everything we know. It is a quiet, automated process that runs in the background of every waking moment, ensuring that our reality has both a place and a pace. This deep biological symmetry suggests that to the human mind, moving through the world and moving through time are essentially the same act. In the stillness of the night, as the body rests, these cells continue their work, refining the maps of where we have been and how long we stayed. The biological foundation of our internal map is a masterpiece of precision, but it is also a delicate physical structure. In the study of neurodegenerative conditions like Alzheimer's disease, we see what happens when the physical architecture of this map begins to change. Scientists look at this process through a strictly structural lens, observing how microscopic alterations in brain tissue lead to changes in how a person perceives their surroundings. This shift in the brain's physical state often manifests first as a subtle change in spatial awareness. One of the earliest and most consistent clinical signs of this biological transition is spatial disorientation. A person might find themselves momentarily confused in a neighborhood they have lived in for decades, or they might struggle to find their way out of a familiar building. This happens because the brain's navigation system is often the very first area to be affected by the underlying pathology. It is not a failure of character or effort, but a direct result of cellular changes in a very specific part of the temporal lobe. The epicenter of this change is typically the entorhinal cortex, the gateway between the rest of the brain and the hippocampus. In the early stages of Alzheimer's, researchers often observe the accumulation of a specific protein called tau. This protein begins to form tangles inside the neurons of the entorhinal cortex, disrupting the cell's ability to function and communicate. Because this region is the primary home of the grid cell network, the damage here has a profound impact on the brain's ability to calculate coordinates. When the grid cells in the entorhinal cortex are compromised by these protein tangles, the brain loses its geometric anchoring. Imagine trying to navigate using a piece of graph paper where the lines are slowly fading away until the page is blank. Without the hexagonal lattice provided by healthy grid cells, the brain can no longer accurately calculate distances or directions. The internal GPS loses its scale, making a short walk feel like an endless journey or causing a familiar corner to look entirely new. This breakdown of the grid network means that the hippocampus no longer receives the high-quality spatial data it needs to create a stable map. Even if the place cells in the hippocampus are still functioning, the coordinates they rely on from the entorhinal cortex are scrambled. This mismatch between what a person sees and what their brain reports creates a sense of being untethered from the environment. Science shows us that this disorientation is a mechanical failure of the brain's internal compass, driven by the physical presence of tau tangles. As the structural integrity of these neural pathways continues to shift, the brain struggles to link new visual landmarks to the old, established maps. The loss of these geometric anchors is a quiet, biological process that reshapes how the world is experienced. By understanding the specific vulnerability of the entorhinal cortex, researchers are better able to identify why the sense of place is so deeply affected. It is a reminder of how much our daily comfort depends on the invisible, rhythmic firing of these specialized cells. Humanity's ancient internal mapping system faces its most significant challenge when leaving the constant pull of the Earth behind. In the microgravity environments of modern orbital stations and lunar missions, the brain must suddenly function without the most fundamental spatial anchor it has ever known. On Earth, the vestibular system in the inner ear uses the persistent downward force of gravity to tell the brain which way is down, providing a permanent vertical axis for every mental map. When an astronaut enters orbit, this reliable signal vanishes, leaving the brain in a state of profound sensory mismatch that researchers often call space adaptation syndrome. This condition occurs because the eyes report movement and orientation, but the inner ear reports a confusing state of continuous freefall. The initial confusion is not just a feeling of dizziness, but a literal breakdown of the brain's coordinate system. Without the heavy anchor of terrestrial physics, the up and right and down that we take for granted become entirely subjective and fluid. Some astronauts describe a phenomenon where they feel as though the floor has suddenly become the ceiling, or that they are tilted at a 90-degree angle despite being aligned with the station's walls. This is the hippocampus struggling to find a stable geometric frame in a world where the gravity-based vertical axis has been deleted. To resolve this, the brain must undergo a rapid and intense remapping process that prioritizes visual cues over the failing signals of the vestibular system. During this adaptation, the structural architecture of the spacecraft becomes the new primary reference for the internal GPS. Astronauts often find that their brains reset their sense of orientation based on where the lights are located or the direction of the text on equipment labels. If an astronaut floats into a module upside down relative to their previous position, the brain may suddenly flip the mental map to match the new visual horizon. This flexibility demonstrates the incredible neuroplasticity of the hippocampal network, which can abandon a lifetime of gravitational rules in just a few days. The brain essentially learns to build a floating map that can be rotated at will, rather than one that is pinned to the center of the planet. This transition into three-dimensional navigation is a monumental shift for a species that evolved to move primarily on a flat, two-dimensional plane. On Earth, our place cells and grid cells operate within the constraints of walking or running across a surface. In the microgravity of a space station, movement involves pitch, roll, and yaw, allowing humans to move through the volume of a room rather than just its floor plan. Studies conducted by NASA and other space agencies suggest that the hippocampus adapts by expanding its mapping capabilities into a truly volumetric, spherical representation of space. This 3D mental lattice allows an astronaut to navigate the complex, cluttered interior of a laboratory module while floating at angles that would be impossible on the ground. As the mission progresses, the brain becomes so efficient at this new form of navigation that the sensation of up or down becomes a choice rather than a physical requirement. This demonstrates that our inner map is not a rigid piece of hardware, but a dynamic software that can be rewritten to suit the environment. The heavy, constant signal of gravity is replaced by a lighter, more agile system of visual landmarks and tactile references. Even in the silent, weightless expanse of an orbital workshop, the mind continues its tireless work of keeping the individual oriented and grounded in their reality. This mastery of 3D space is a testament to the resilience of the human nervous system in the most extreme conditions imaginable. The lessons learned from the stars eventually find their way back to the tools we use on the ground. Modern technology has created a unique challenge for the ancient navigational hardware inside our skulls by introducing virtual reality. When a person puts on a headset, the eyes perceive a vast, three-dimensional landscape while the body remains stationary in a chair or a small room. This creates a fascinating sensory conflict that neuroscientists are only beginning to fully decode. The brain must decide whether to trust the high-definition visual data of a digital world or the physical signals from the inner ear and muscles. It is a testament to the flexibility of the hippocampus that it can build a functional map of a place that does not technically exist. Researchers have conducted extensive experiments to see how individual neurons react to these synthetic environments. In these studies, rats or human subjects navigate digital mazes while their neural activity is monitored with incredible precision. The results show that place cells do indeed fire in virtual reality, creating a mental representation of the digital space. This suggests that the brain is willing to accept visual depth cues as a primary source of spatial information. Even without the physical sensation of walking, the mind tiles the virtual floor with the same cellular accuracy it uses in the real world. However, the internal map is not identical to the one created during physical movement. One of the most striking differences involves the rhythm of the brain itself. In a natural environment, movement triggers a steady, rhythmic pulse of electrical activity known as the theta wave. This wave acts like a conductor, synchronizing the firing of grid cells and place cells to ensure the map stays updated in real time. In virtual reality, because the body is not physically moving, this theta rhythm becomes significantly altered and less stable. The lack of proprioception, the sense of one's own body position, means the brain is working with an incomplete data set. While the visual cortex is fully engaged by the light and shadows of the simulation, the vestibular system in the inner ear remains quiet. This mismatch can sometimes lead to a breakdown in the spatial network's precision. Without the step-by-step confirmation of physical motion, the grid cells may struggle to calculate distances with their usual mathematical perfection. Despite this, the human brain remains remarkably adept at navigating these illusions, proving that our internal GPS is a master of adaptation in the face of artificial reality. The human brain does not require light to construct a perfect geometric model of its surroundings. While many people assume the internal map is a visual product, it is actually a multisensory construction that can flourish in total darkness. For individuals who are blind, the hippocampus and entorhinal cortex do not sit idle. Instead, they recruit every available stream of information to tile the world with grid and place cells. This remarkable adaptation proves that our sense of space is a fundamental biological framework rather than a simple byproduct of sight. One of the most extraordinary ways the brain achieves this is through human echolocation. By producing sharp clicks with the tongue or tapping a cane, a person sends sound waves out into the environment that bounce off physical objects. These returning echoes carry precise data about the distance, size, and density of everything in the vicinity. Recent neuroimaging studies have revealed that when an expert echolocator processes these sounds, the visual cortex in the back of the brain becomes highly active. Even though no light is entering the eyes, the brain repurposed its visual hardware to process spatial echoes, effectively seeing the world through sound. This auditory data allows the brain to calculate the exact geometry of a room with startling accuracy. A person might feel a subtle change in air pressure as they walk past a large pillar or sense the acoustic shadow of a nearby doorway. These tiny sensory inputs are fed directly into the spatial navigation network, where they are transformed into a stable coordinate system. The brain can distinguish between the hard, reflective surface of a glass window and the soft, absorbent texture of a heavy curtain just by how sound behaves in that specific pocket of space. Tactile feedback also plays a vital role in grounding these non-visual maps. Every step taken on a tiled floor versus a plush carpet provides a distinct data point that the speed cells and border cells use to calibrate the map. This constant flow of touch and vibration acts as a tether, ensuring that the internal representation of the house or city remains locked to the physical world. It is a slow, methodical building process that results in a cognitive map just as detailed and reliable as one built through vision. The sheer power of neuroplasticity is on full display during this mapping process. The brain demonstrates a tireless ability to reroute functions and maximize its resources to ensure the organism can navigate safely. This confirms that the hippocampal map is a universal feature of the human experience, a biological necessity that exists independently of any single sense. Whether the input is light, sound, or touch, the result is the same: a sophisticated three-dimensional understanding of where we are and where we are going. Science for sleep often reminds us that our brains are always working to keep us oriented, even when our eyes are closed for the night. This deep-seated need for a map is what allows us to feel secure in our environment, knowing that every corner and hallway is accounted for in our mental architecture. The resilience of the inner GPS is a testament to the brain's priority for spatial awareness. Even without the sun or a lamp to guide the way, the mind remains a master architect of the spaces we inhabit. The relationship between what we smell and where we are is one of the most ancient partnerships in the history of the animal kingdom. Deep within the temporal lobe, the olfactory bulb sits in physical and functional proximity to the hippocampus, forming a direct neural highway between the nose and the brain's mapping center. This evolutionary link is far from accidental, as the very first brains likely evolved not to see the world, but to navigate it through a shifting landscape of chemical signals. In many primitive organisms, the primary function of the early cortex was to process odors to find food or avoid danger. Because of this heritage, our modern brains still treat a scent as a high-priority coordinate for our internal GPS. Olfactory navigation operates on the principle of gradients, where the intensity of a smell acts as a compass needle. While humans often believe we rely almost exclusively on our eyes, We are constantly and subconsciously sampling the air to verify our location. Animals use these invisible plumes of molecules to build complex, three-dimensional maps of their territories, tracking how a scent becomes more concentrated as they move toward its source. Even for us, the subtle change in the air when moving from a pine forest into a grassy meadow provides a spatial boundary that the brain uses to update our position. These chemical landmarks are incredibly stable over time, providing a layer of environmental data that visual landmarks simply cannot match. When a specific odor enters the nasal cavity, it can instantly snap a dormant mental map back into sharp focus, a phenomenon often referred to as the Proustian effect. This happens because the olfactory system bypasses the thalamus, the brain's usual sensory relay station, and sends signals directly to the amygdala and the hippocampus. When these regions receive a familiar chemical signature, they don't just recall a single fact. They reactivate the entire spatial network associated with that smell. A single sniff can trigger a flood of spatial memories, causing the brain to rebuild a room, a street, or a landscape that has not been visited in decades. This rapid reactivation proves that our internal maps are not just made of geometry, but are deeply infused with the chemistry of our experiences. Consider the specific, sharp aroma of rain hitting hot pavement on a summer afternoon, a scent known as petrichor. You might be standing in a completely different city, yet the moment that scent reaches you, your brain may instantly orient you back to the specific childhood street where you first experienced it. You can suddenly visualize the exact height of the curb, the position of the neighbor's fence, and the way the sidewalk slanted toward the storm drain. This is not just a vague memory, but a precise spatial recalibration triggered by a few molecules of earth and water. The brain uses that olfactory anchor to pull an old, dusty map out of storage and overlay it onto your current reality. Science for sleep often highlights how these background processes provide us with a quiet sense of continuity throughout our lives. Even as we drift off, our brain remains sensitive to the familiar scents of our home, using them to confirm that we are in a safe, known location. This constant, quiet monitoring of the chemical environment ensures that our mental map remains locked to our physical surroundings. It is a comforting realization that our sense of place is rooted in something as fundamental and enduring as the air we breathe. Every breath we take is a data point, a gentle reminder from the hippocampus that we are exactly where we belong. Human spatial navigation is not a single, uniform process, but rather a collection of different internal strategies that our brains employ based on the environment and our own biological predispositions. Researchers in the fields of psychology and neuroscience have long observed that individuals often favor one of two primary methods for finding their way through the world. These methods are categorized as either allocentric or egocentric navigation, and each relies on distinct neural pathways within the brain's mapping system. Understanding these two frameworks helps clarify how different people can look at the same physical landscape but process their position within it in entirely different ways. The allocentric strategy is often described as a world-centered approach to navigation. When you use this method, your brain builds a mental representation of the environment that exists independently of your own body's current position or facing direction. This involves thinking in terms of cardinal directions, such as north and south, or using the global geometry of a space to stay oriented. If you find yourself thinking that a specific building is located on the far eastern edge of a park, or that a mountain range serves as a fixed northern border for a valley, you are utilizing an allocentric map. This strategy is highly flexible because it allows you to plan new shortcuts or navigate through unfamiliar territory by relying on a stable, objective grid of the world. In contrast, the egocentric strategy is a body-centered approach that focuses on the navigator's personal perspective. This method relies heavily on sequences of movements and specific local landmarks to guide the way. An egocentric navigator remembers a route as a series of specific actions. Turning left at the red brick house, walking straight for 3 blocks, and then turning right at the pharmacy. This strategy creates a reliable, step-by-step path through an environment without necessarily requiring an understanding of where those points sit on a global compass. It is an incredibly efficient way to move through familiar routines, as the brain can almost automate the process of traveling from one point to another by recognizing familiar visual cues along the way. Biological and psychological studies have frequently explored whether there are consistent differences in how men and women utilize these two strategies. Some data suggests a general trend where men may more frequently lean toward allocentric strategies, prioritizing global geometry and cardinal directions to find their way. Conversely, some research indicates that women may often favor egocentric strategies, showing a high degree of proficiency in remembering specific landmarks and complex, multi-step routes. These observations have led to many discussions about the evolutionary origins of these preferences, perhaps relating to ancestral roles in wide-ranging hunting or local gathering. However, it is important to note that these findings are often contested within the scientific community. While some studies show clear statistical differences between groups, others find that the variation within a single gender is often greater than the variation between them. Many researchers argue that these preferences are not hardwired biological imperatives, but may instead be influenced by cultural training, early childhood play, or the specific demands of a person's daily life. There is a healthy debate about whether these strategies are truly distinct or if they exist on a broad spectrum where most individuals use a hybrid of both methods. This nuance ensures that we avoid oversimplifying the complex reality of how a human brain interacts with its surroundings. Regardless of which strategy an individual might favor, the human brain is remarkably adept at switching between them seamlessly. You might use an egocentric route to drive to a new part of town, following a list of turns and street signs. But if a road is blocked by construction, your brain can instantly shift into an allocentric mode, visualizing the broader layout of the city to find an alternate path that eventually leads back to your destination. This cognitive flexibility is a hallmark of the hippocampus and the surrounding cortex, which work together to ensure we are never truly lost. Both strategies are equally effective for surviving and thriving in a complex world. The egocentric method provides a high level of detail for navigating the immediate, lived-in environment, while the allocentric method offers the big-picture perspective needed for exploration and large-scale planning. As you rest tonight, your brain continues to manage these two perspectives, filing away the routes you took today and reinforcing the landmarks that will guide you tomorrow. This constant, quiet coordination between our internal compass and our memory of the world is what allows us to move through life with confidence. Understanding these relaxing facts about our mental maps can make the world feel a little more navigable and the mind a little more settled. The rise of modern satellite navigation has fundamentally altered how we interact with the physical world, creating a silent shift in our neural workload. For most of human history, moving from one point to another required a constant, active engagement with the environment. We had to pay attention to the position of the sun, the slope of the land, and the specific sequence of landmarks that signaled a correct path. This active processing kept the hippocampus in a state of high engagement, as it was forced to build and update a complex internal map in real time. Today, much of that cognitive heavy lifting has been outsourced to the glowing screens on our dashboards and the smartphones in our pockets. When we follow turn-by-turn digital instructions, we often enter a state of passive navigation where the brain no longer needs to calculate its own coordinates. Recent neuroscientific studies have utilized brain imaging to compare these two distinct modes of travel. Researchers found that when people navigate manually using their own spatial memory, there is a significant spike in activity within the hippocampus and the prefrontal cortex. In contrast, when those same individuals follow the automated prompts of a GPS, these specific brain regions show remarkably low levels of activation. It appears that by following a blue line on a screen, we effectively put our internal mapping system into a standby mode. This shift brings us to the core principle of neuroplasticity, often summarized by the phrase, "use it or lose it." The brain is a highly efficient organ that allocates its limited energy and physical space based on demand. Just as a muscle might atrophy if it is no longer challenged, the neural pathways responsible for spatial awareness can become less dense over time if they are rarely engaged. Longitudinal observations suggest that relying exclusively on automated navigation may lead to a measurable decrease in the volume of gray matter within the posterior hippocampus. This is not necessarily a defect, but rather a biological adaptation to a world where high-level spatial calculation is no longer a requirement for daily survival. Viewing this change neutrally allows us to see it as a trade-off in our evolutionary journey. By offloading the taxing work of navigation to technology, we free up mental energy for other complex tasks, such as creative problem-solving or social interaction. However, it also means that our innate sense of direction may become less sharp without intentional practice. Understanding these relaxing facts about our changing brains helps us appreciate the incredible flexibility of our biology. Even in an age of digital maps, the potential for a robust internal compass remains within us, ready to be reactivated whenever we choose to look up from the screen and truly observe the path ahead. Such reflections on our modern habits provide a calming perspective on how we continue to evolve alongside our tools. Deep within the architecture of the brain, a rhythmic pulse governs the way we move through the world. This internal beat is known as the theta wave, a slow and steady electrical oscillation that flows through the hippocampus. These waves typically vibrate at a frequency of 4 to 8 Hz, creating a consistent background hum for our spatial thoughts. When we begin to walk or run, these waves immediately spring into action, providing a temporal framework for our navigation. They act as the primary clock of the inner mind, ensuring that every signal sent by our neurons is perfectly timed with our physical motion. This biological rhythm is essential for keeping our internal maps updated in real time as we traverse different environments. Think of these waves as a quiet, rhythmic metronome that guides the symphony of our brain cells. For the grid cells and place cells to work together, they must fire in a specific sequence relative to one another. The theta wave provides the essential beat that allows these different types of neurons to synchronize their activity. Without this steady pulse, the data from our speed cells and head direction cells would likely become a chaotic jumble of information. By locking into the theta rhythm, the brain ensures that the map of where we are stays perfectly aligned with the map of where we are going. It is a masterpiece of natural engineering that allows for seamless travel through complex spaces. The speed of these oscillations is not fixed but instead shifts based on how fast we are moving through our surroundings. When we pick up our pace, the frequency of the theta waves tends to increase, reflecting the higher demand for rapid data processing. Conversely, as we slow down to a leisurely stroll, the waves become longer and more drawn out. This relationship between physical speed and neural rhythm allows the hippocampus to maintain a precise record of distance traveled. Even when we are simply navigating a familiar hallway, this quiet electrical tide is constantly rising and falling in the background. It is the fundamental language of movement, translating the physical act of walking into a digital-like code the brain can understand. As we eventually come to a halt and prepare for a period of stillness, a remarkable transition occurs within these brainwaves. The brisk, active theta rhythm begins to fade, giving way to different, even slower patterns of electrical activity. This shift signals to the rest of the brain that the period of active exploration has ended and the time for processing has begun. The once sharp metronome softens, allowing the neural environment to settle into a state of quiet reflection. This natural deceleration is a key part of how the body transitions from the alertness of the day into a restorative state. It is as if the internal map is being folded up and put away for the night, leaving the mind in a state of calm. Watching this transition through the lens of science reveals how deeply our movement is tied to our rest. The same structures that help us find our way through a forest also help us find our way into a deep, dreamless sleep. When the theta waves subside, they often make room for the gentle ripples of memory consolidation that occur while we are stationary. These slower frequencies create a sense of internal distance from the demands of the external world. By understanding these rhythmic shifts, we gain a clearer picture of how the brain manages its energy and focus. The steady pulse of the hippocampus eventually yields to the silence of a mind at peace, marking the end of the day's journey. The human brain possesses an incredible capacity for adaptation, taking the ancient machinery used to navigate physical landscapes and applying it to the vast world of abstract thought. This transition represents one of the most significant evolutionary leaps in our biological history, as the same neural architecture that once tracked the path to a watering hole now tracks the flow of a complex logical argument. Scientists have begun to uncover how the hippocampus and the entorhinal cortex, the traditional centers for spatial mapping, are recruited to organize non-spatial information into structured mental frameworks. This process allows us to categorize experiences and ideas with the same geometric precision we use to understand a physical room. Recent neuroscientific theories suggest that grid cells, those remarkable neurons that create a hexagonal lattice over our physical surroundings, are also used to organize conceptual knowledge. Instead of mapping latitude and longitude, these cells may create a coordinate system for abstract dimensions, such as the varying traits of a social hierarchy or the chronological steps of a long-term timeline. When we learn a new subject, our brain may be literally tiling that field of knowledge with a grid, allowing us to see how different facts relate to one another in a structured mental space. This internal scaffolding provides a sense of distance and direction within our own minds, making it possible to recall information by moving toward it. This internal mapping extends even into the realm of sensory experiences like music, where the brain treats changes in pitch as movements through a specific kind of acoustic space. Research published in journals like Nature and Science indicates that the same hexagonal firing patterns seen during navigation are present when humans mentally navigate through ranges of sounds or bird silhouettes with varying neck lengths. This suggests that the brain does not have separate systems for physical maps and intellectual concepts, but rather uses one universal engine to make sense of any data that has a logical order. By placing abstract ideas onto a grid, the mind can calculate the distance between two thoughts or the most direct route to a specific memory. Our daily language is filled with evidence of this deep biological connection between space and thought, often without us even realizing it. We frequently speak of navigating a difficult conversation or reaching a conclusion as if we were physically traveling toward a destination. When we find ourselves lost in thought or feel that an argument is going nowhere, we are using spatial metaphors that reflect the actual activity occurring within our hippocampal circuits. This linguistic habit is not a coincidence but a direct reflection of how our brains are wired to process information as a series of locations and paths. The way we perceive social relationships also relies on this ancient navigational hardware. As we map out close friends and distant relatives within a social space, we might visualize someone as being above or below us in a professional hierarchy, assigning them a specific coordinate in a mental landscape of status and influence. This ability to see social dynamics as a physical layout helps us predict behavior and navigate the complexities of human interaction with greater ease. The brain effectively creates a social map that functions almost exactly like a map of a neighborhood, complete with landmarks and well-worn paths of interaction. As we consider how deeply these maps permeate every aspect of our existence, it is fascinating to wonder if all human thought is simply the act of navigating through an invisible, multidimensional geometry. This perspective suggests that our most profound intellectual achievements are rooted in the simple, ancestral need to know where we are. By repurposing our spatial cells for high-level cognition, the brain has turned the entire universe of ideas into a territory that can be explored, charted, and understood. This elegant system ensures that no matter how abstract a concept becomes, we always have a familiar way to find our way through it. The human brain possesses an incredible capacity to transform the chaotic signals of the external world into a coherent and stable internal landscape. This journey of discovery began with the simple observation of a single neuron firing in a specific corner of a room, a finding that revealed the existence of place cells. From those humble cellular origins, we have come to understand a sophisticated biological architecture that includes hexagonal grid cells, directional compasses, and boundary markers. These components work in perfect harmony to ensure that we are never truly lost, even when the lights go out or the path ahead becomes uncertain. Science has shown that our internal GPS is not merely a tool for finding our way home, but is the very foundation of how we experience our lives. By anchoring our memories to specific coordinates in space, the hippocampus allows us to build a rich and detailed history of our personal experiences. We do not just remember what happened. We remember where we were, the way the light hit the walls, and the distance between ourselves and the people we love. This spatial scaffolding turns a collection of random moments into a meaningful narrative that we can revisit whenever we close our eyes. The transition from mapping physical rooms to mapping abstract concepts represents one of the most significant leaps in evolutionary history. It is a testament to the brain's efficiency that it uses the same neural hardware to navigate a forest as it does to navigate a complex mathematical problem or a social hierarchy. This realization suggests that our intellect is deeply rooted in our physicality, and our highest thoughts are still guided by the ancient geometry of the natural world. Every idea we hold has a place in the vast, invisible territory of the mind, waiting to be rediscovered. Even in moments of complete stillness, the hippocampus continues its quiet and essential work in the background of our consciousness. While we rest, this dedicated region of the brain is busy replaying the day's journeys, strengthening the connections that form our long-term knowledge. It sifts through the data of our movements, discarding the trivial and highlighting the paths that will be important for our future. This process of consolidation ensures that when we wake, our internal map is updated, refined, and ready to guide us through another day of exploration. As the pace of the world slows down, it is comforting to know that our internal sense of place remains a constant and grounding force. The intricate lattice of grid cells and the steady firing of head direction cells provide a sense of stability that persists regardless of our external circumstances. This biological certainty allows us to let go of the need for active navigation and find peace in the present moment. We can trust that our mind has already charted the territory, leaving us free to simply exist within the safety of the maps we have created. The study of the brain's navigational systems reminds us that curiosity is a fundamental part of being alive. Whether we are exploring the streets of a new city or the depths of a scientific theory, we are engaging in a process that is millions of years in the making. Understanding these relaxing facts about our own biology can help us appreciate the quiet genius that operates within us every single second. This internal guidance system is a gift of evolution, a silent partner that keeps us oriented in an ever-changing universe. If you have enjoyed this peaceful look into the wonders of neuroscience, please consider subscribing to the channel for more science for sleep. The perfect map of the world is already contained within you, waiting to be explored in the quiet moments of deep curiosity and restful sleep.